talking about aging genes and how we actually can work on aging and use different approaches to actually find out more about aging. I suppose you can hear me okay? Yes. Sorry. Okay, great. Thank you, Bernard. Perfect. Okay, so how do we age? Do we have aging genes? Can we pinpoint aging genes apart from the ones that we already know, which are the ones that are involved in all these progeroid syndromes? Can we find ways of actually going about the genome and finding out where are these aging genes? Okay, so we can do that by observing aging rates. So we can use, mainly for this, we need to start having animal models. And so if we want to have do experiments, doing genetic experiments, observing aging, changing conditions, and then observing aging rates, we need, in fact, to use animal models because we cannot really do experiments with humans. Okay, with humans, we can study genetics by looking at genetic variants, and we can observe different environments if we're studying population genetics. But in order to go really pinpoint and start doing real experiments, we really need to have model organisms. And so we need to observe aging rates in different organisms. And we can do something which is a genetic screen. We can mutate many different organisms and then select the ones that are living longer. This is the principle of doing this classic genetic approach of a genetic screen. We just mutate a high number of individuals, and with this, then we can identify lifespan determining genes, we can look at the proteins, we can study the biochemistry, we can study the cells, we can do many different things, okay? But all of this we can then be doing in the lab. So one of the first people that started looking at aging rates in organisms was Raymond Pearl, and Raymond Pearl was working with Drosophila. And Drosophila melanogaster, as all the people that work with Drosophila know perfectly well, this came from many different experiments in the 20s, is that if we put Drosophila at higher temperatures, around 25 degrees, the life cycle is 10 days. If we put them at lower temperatures, like 18 degrees, then life cycles again expanded. And not just life cycle is expanded, but also lifespan is expanded. So what Raymond Pearl observed is that if you put flies at 18 degrees, they would have a maximum lifespan of beyond 150 days, whereas if you had them at about 27 degrees, would that have about a third of what you have at 18 degrees. And so he formulated this theory, which was called the rate of living theory. The duration of life varies inversely as the rate of energy expenditure. The length of life depends on the rate of living, which is in fact a little bit a consequence of what was happening to Luigi Carnaro when he was living fast and having lots of partying, eating lots and drinking lots. His life, then when he started slowing down the energy expenditure, then he became living longer. But in this case, in the rate of living theory, Raymond Pearl was based only in temperatures. And so from this, we could conclude that at lower temperatures we live longer. And so there was lots of theories coming out after this, and you probably know a few of these, that if you go into a cold chamber, then you're going to live longer. And there's lots of companies now selling treatments in cold chambers, so then you are supposedly living longer. Obviously, this is a rate of living theory that was based on the temperature in insects like Drosophila melanogaster that cannot mandate their body temperature. So their body temperature depends on the outside temperature. If we put them at 18 degrees, all their cells are at 18 degrees. If we put a fly at 27 degrees, all of the cells inside the fly are at 27 degrees. And so this is the main difference between insects and mammals. We can maintain our body temperature, so we can go into the fridge, and we are not going to be changing our lifespan. In fact, we might, but the other way around, because we might. And so the idea is that if you have cells or if you have organisms where you can lower their temperature, energy expenditure goes down because cells will then be, everything will slow down, and then you can increase lifespan. This is not very, the temperature part is not very useful for mammals, but we can use this as a study of energy expenditure. So the lower energy you use, in this case, your cells use, the longer you might be living. And then we have other ways of looking at cells. So we have the aging rates, we observe aging rates, but people also are very interested in finding out what happens to cells. And so Leonard Flick was doing experiments to see what was the maximum number of times that cells would divide. And so he manipulated them, and what he could see is that replication, so division of these cells, slows and stops after about 50 passages, 50 plus or minus 10. So if you are doing experiments with cells and they divide and divide and divide, and they have an exponential growth phase, if you continue making them divide by passing them, you will reach a point where they don't divide anymore. And this part was also called by Hayflick as the senescence. And all of this was then called the Hayflick limit, and it's still called the Hayflick limit. Now we know that cell replication slows and stops, and so if we have cells that are normal, it's impossible to keep them going in the lab for more than 50, 60 passages. So we know that they enter senescence, and this will mean that then our cell line is not able to divide anymore. So the only cell lines that we have dividing immortal in the lab are the ones that have been transformed, and many of them have been transformed with viral agents, and some of them are actually cell lines that are derived from cancers, and these ones, yes, they can continue dividing, but cells that are normal, they will enter senescence. So the Hayflick limit was telling us many things about the cells, okay? So all the cells that are dividing, they will only be able to divide 50 times, and then they will stop. But then again, we go back to the poll that I was giving you before. Most of the cells in our body, the majority of cells in our body at any given time are not dividing. So this 50 passage limit, this 50 division limit, is actually not going to be affecting most of the cells in our body. Okay, so then people started later on, and I started already talking about in the 80s, searching for aging genes. Okay, so we have all these observations. We know that if we put things in lower temperature, they will live longer. If we put individuals at higher temperature, they will live less. We know that cells will stop dividing after 50 times, but we really need to go and look at organisms and see what happens. And so in the lab of Michael Klaas, they did the first screen for long-lived mutants. And so what they did is they used nematodes. They use C. elegans, nematodes that you have here. This is a C. elegans at day 5, and this is a C. elegans at day 16. So that as model organisms, they're very good for study aging because you don't have to wait for very long. You know that you're young at day 5 and you're old at day 16. And so you can easily think about the strategy, which is a genetic screen where you mutate many different individuals. These individuals that are mutated will create progeny which is mutated, and then you select the ones that when they reach day 16, they don't look like this and they don't have all the characteristics of an older. So you can select not just lifespan, the ones that live longer, but you can also select the ones that have a higher health span, so the ones that are living healthier for longer. And so they did this, obviously, because it's much better to select for long-lived mutants than to select for short-lived mutants. So a little bit the two coins, the supercentenarians and the progeroid ones. Okay, so what are the conclusions in this screen? So the first screen was done with C. elegans, and Michael Klaas, in the lab of Michael Klaas, this is his first screen, and then a few years later, they identified AGE1, which was one of the mutants that came out of this screen, as a PI3 kinase mutation. If you had a mutation in AGE1, these worms would have double lifespan, so they will increase 110% their maximum lifespan. So you would have worms that would then live much longer than the other ones, but very importantly, you had these worms remaining youthful for longer. So these worms were not just living longer, but they were looking younger and moving as younger nematodes for much longer than the ones that were wild type. They could also show that this is a mutation that was passed on to the different generations. So you had AGE1 mutants, and if you could pass this AGE1 mutation from generation to generation. And so they demonstrated that increase in lifespan was heritable. So there was a genetic component to it. Then a few years later, based on the same mutant, Cynthia Cannon and the lab of Cynthia Cannon isolated mutations in DAF2. And DAF2 were different worms that would be also increased in their lifespan, and they also increased in their health span. And I will show you a video on that in a couple of slides. But from this, you started sort of opening the field of genetics of aging. And so many studies in many other different model organisms were done in order to discover more genes. So what happened? All the genes that were found in the years around in the 90s, up to the end of the 20th century, from all these genetic screens that most of these genes are related to the maintenance and integrity of DNA. So we go back to what we were talking before. Many of these genes were related to DNA repair. So if you had mutations that made the DNA repair machinery work better, you would get mutants that would live longer. But then some C. elegans genes were related to the ability to enter Dower status. Dower of the C. elegans worms, when they are subjected to nutrient deprivation, they enter this Dower status where it's a quiescent animal status where they require the minimum energy and they can be quiescent for a while until the food goes up again and then they can enter normal status again. And so many of these genes that were selected as one that then increased lifespan, and then they increased lifespan not because the worms were in Dower state, but because they were in a normal state, they would still increase lifespan. They are related to nutrient sensing pathways. Okay, so if you have a mutation in some of the genes which are involved in nutrient sensing pathways, which then will also are involved in entering Dower status, you are able to make C. elegans live longer. And so this is a small cartoon of that. So you have DAF2 mutants and AGE1 mutants that we talked about already. They usually switch on the Dower longevity program in adults. So you have a normal adult. If these nutrients go down very abruptly, this one will enter the Dower state. But then if you have mutations in DAF2 and in AGE1, this mutant adult will live much longer than the other ones, so the ones that don't have the mutations. These mutants will continue living and have a much healthier status than the ones that are not mutants for these genes. Okay, so this mutation, we select this mutant, and then we need to find out where the mutation is in which gene and what is this gene. And so first I will show you this video where Cynthia Cannon is talking, but I will make her do it in silence because it's not necessary. And so this is Cynthia Cannon explaining us what the worms look like. This is a normal worm. It moves normally and it's crawling. And so you can see it here in the next frames. You have this worm. It's just crawling around. This is a young one. And so then what they did is they wanted to look at the ones that came out of the screen. And so they looked at the ones that came out of the screen were mutants and looked at them when they were young, and they looked exactly normal. So they were mutants, but in younger stages, they moved and they had all the characteristics of a normal worm. But then they looked at the older wild type. So normal worms that were 13 days old, they looked like this, and you saw a picture before. They start losing integrity and they cannot move as the others. So they're just kind of impaired and they have problems moving, and they show all the aging types that you can see in a worm. In fact, this one here, you can hardly see it, and this one here is only moving a little bit. So you can see that these ones are nearly dead. But then you look at the 13-day-old adults of the long-lived mutants, and they look exactly like young adults of non-mutants. And so not only they could isolate mutants in DAF2 that were able to live longer, but they could live longer healthier. And this was what the important part that we were talking about at the beginning. Oh. There was a problem with the sound. Okay. So this is the conclusion. You can, by a single mutation gene, you can increase lifespan and you can increase health span. And so, okay, at the beginning, people were saying, okay, this is great, but this is only to do with C. elegans because C. elegans have this Dower state, and this is why we have all these nutrient sensing pathways, until it was found that all these C. elegans lifespan genes have human homologs. And so AGE1 is homolog of PI3 kinase, as I was saying before. DAF2 is a homolog of the insulin of the IGF-1 receptor, and DAF16 is one of this transcription factor called FOXO, which is downstream of the insulin receptor. And all this at the same time, not just the homologies with human genes, but also the universality of all of these has made the nutrient sensing pathway one of the axes of aging research. And so it was shown that you could have the effects in worms, in flies, in mouse, and in humans, that a single mutation in the receptor for the insulin, which is represented here, and it's called DAF2 in worms, insulin receptor in flies, IGF-1 or insulin receptor in humans, a mutation here that impairs the signaling downstream is able to increase the lifespan of these organisms. Okay, so what is the effect of this mutation? The effect of these mutations in DAF2 or in the others are the same as not having this ligand here. And what is this ligand here? If this is the insulin receptor, this is insulin, and this is what detects how many calories you're ingesting, more or less. And so what happens is that if we lower the amount of insulin in the body, and if we have less signaling from this receptor to inside of the cell, we are going to create conditions where the animals can live longer. So we go back to Luigi Canaro, and we go back to all the dietary restriction data that had been done before and nobody could really explain. And so genetically, we arrived at the same conclusion that people had been arriving doing dietary restrictions for many, many years before. If we lower the ligand here, or if we make a mutation in this receptor, the response downstream is a response that leads to a longer lifespan. And so if we are then going to meet Luigi Canaro 500 years later, like now, we could tell him that, you are right. If you're doing caloric restriction, you're doing that. And so you're inflicting a difference in the downstream effects of the insulin receptor pathway. And therefore, this is why you're going to be living longer. We also know this not just by studies in the worms and in the flies, but we also know that calorie restriction, not just Luigi Canaro, but this is also other studies that have been done. If you reduce calorie intake in many different organisms, you increase lifespan. If you introduce mutations in nutrient sensing, you can extend lifespan, in some cases by up to 50%. In the case of the worms, up to 100%. These supercentenarian mice, because they live much longer than that, they have delayed or reduced age-related diseases. So they have increased lifespan and increased health span. And this is all also related and not different with what was discovered 500 years ago by Luigi Canaro. So the difference is that we can now pinpoint this genetically, because of course you can do dietary restrictions, and I bet that you all tomorrow are going to stop eating so you can live longer, but that's not the idea. Because of course, if you go into very seriously into dietary restrictions, you're going to run into different problems as well. Because of course, people that go very serious restriction, they start having osteoporosis problems because they don't ingest enough calcium. They start having heart rate problems because they have too very sugar levels that are very low, etc., etc., etc. So the idea is that you find genetically ways of dealing with the nutrient sensing pathways that are not affecting directly the ingestion of calories by people. Okay, so this is a summarized version of what we know now. It's a lot more complicated than this. What about fats? Okay, we're getting that, Laura. We're getting to there. So this is what we have here. We have nutrient sensing pathways. We have the insulin, which is more sugar, but then we have been finding years that it's not just sugars and insulins, but we also have fats, and this is what on the side here with leptins and the signaling from fats. And then you also have from the other side here signaling from amino acids. For instance, that feeds into the TOR pathway. But if you focus on the middle here, this is the insulin one, which is the one that was found before. Just so you know, you understand a little bit about what's going on, because this is very complicated. And in fact, it's much more complicated than this. There's many different relationships between receptors and ligands, and then cascades. You have PI3 kinase, then you have another protein called AKT, which is in the center of all this because it makes all these different pathways interact. And then you have FOXO, which is a transcription factor that is directly downstream the insulin receptor. But you also have other transcription factors that are all stated here. Okay, so the idea is not that you actually know all of this, but that you know that there are different ways of sensing caloric intake, and caloric restriction will lower this. So caloric restriction will lower insulin, caloric restriction will lower this, and this will make the different signaling pathways behave in a different way. For instance, if you think about one of the drugs that is now used for people that are diabetic, not the ones that are injecting insulin, so not the ones that actually have diabetes type 2. These people take pills which are based on metformins, and metformins are feeding into these pathways. And so you can actually touch the pathway and make people, because in this case that they have diabetes, push lower their levels, and they stop being diabetic, or they stop having the lowers of sugar levels in the blood that they have, because you're changing the way they see biochemically all these pathways. Okay, so let's focus in the middle here. You have the insulin receptor or the insulin growth factor receptor, and this senses mostly sugars because it's the insulin. And then you have downstream of this a pathway, which is this is direct, this one activates this one activates pituitary, which then inhibits FOXO. If you inhibit FOXO, what will have downstream of here is that FOXO is going to be downregulated, because the more insulin you have, the more downregulated FOXO is, and you will impinge less on longevity. And I have a cartoon here, so that makes it easier for us to see. So if we have lower activity of the insulin receptor, because we have less sugar intake, or because we have a mutation in the insulin receptor, that will increase longevity of organisms. Okay, this is a direct thing that we saw. A single mutation in DAF2 in worms, which is the insulin receptor in worms, will make these worms live longer. Okay, lowering sugar intake will make organisms live longer. It's the same effect. You lower sugar intake, you lower the activity of the insulin receptor. What will happen then? If you have this, you have higher amount of FOXO, and FOXO is a fork head transcription factor. This is O of the family, because you have A, B, C, etc. And as FOXO, as the insulin receptor activity goes down, this inhibition is not done, and therefore FOXO increases. And as FOXO increases, it goes into the nucleus and does its transcription factor things, and this will increase longevity. But of course, up to here, we know exactly what's happening. But then what happens inside the nucleus is still being studied. There's many studies being done now that are seeing what exactly is FOXO regulating, which then leads to longer lifespans. Okay, so it's not clear FOXO is a transcription factor and is touching many different things. Okay. What change in this pathway would decrease longevity? Well, in this case, you don't really decrease longevity. So this will only, the changes that we can inflict in this pathway are for increasing longevity. There is, as far as I know, not known any mutation in this that makes a progeroid syndrome. So there's no individual with a mutation or a worm with a mutation in a pathway like this which will age faster, if that was your question. Okay, so this one inflict in increasing longevity. Why? Because it's actually very complicated if you think about it, because you're dealing with all the nutrient sensing capacity of a cell. So the cell is sensing the nutrients, and this, if you lower them, you will increase longevity. But if you increase them, you're not going to, of course, you will develop other things like diabetes, etc., etc. But it's not as linear as that. So it's not like DNA repair, many of the DNA repair pathways that you have a mutation and you have people that age very, very fast and they live less. Okay? Okay. So the same thing will happen to many of these. So if you have an inhibition, you have this transcription factor, this will lead to autophagy. Many of these are regulated in ways. Less insulin receptor does result in other pathologies, which are exactly. So you have to have balances, and this is why, okay, you can deal with caloric restriction, and even that is very complicated. But if you start touching the elements of this, which is a simplified version pathway, you are going to have consequences which have to do with the imbalance on other parts of the pathway. So which is why you can think about mutations, and mutations in worms are very easy to study, because you have a worm, you mutate the insulin receptor, you have this insulin receptor mutation, and therefore you're going to live longer. But then what happens to humans? You cannot use brute force in order to increase lifespan. You have to look at small variants that change the pathway in a way that you can actually then see if they're increasing lifespan or not. What about the other? Yes, of course. I mean, if you think about this, you're talking about many of these are environmental factors, because caloric intake is an environmental factor. The more calories you eat is actually an environmental factor. So then you have other environmental factors that not. And for instance, one of the studies that is being done now is to actually study metabolism associated with exercise, so with sport, and seeing how this sport then feeds into the nutrient sensing pathway in a way that in the muscles you can study if you have less or more insulin receptor stimulus, and therefore if you can increase lifespan with this environmental effect of exercise. So there's many different ways of looking at this. And also, I don't want you to think that, and we will get into that in a few minutes, that aging is only caloric restriction or DNA repair. There's a lot more to it as well. Okay, so this is only an example, and this is a very nice and elegant and historic and good example, and especially for you that you're biomedical students, that you have of touching something genetically and being able to increase the lifespan of an organism. So you have the two cases where it's actually very clear. You have a mutation in one gene, in the case of DNA repair, and these people age faster. And here you have mutations in one gene, and these people will age slower and increase their lifespan just with one single mutation. Okay, so this is the mutations that we have in the worms. Now we're going to see what happens in humans, which is not as clear as that. Also because we're not worms. Second, it's not so easy to do experiments. So what can we do with humans? With humans, we can study variants, and variants are the polymorphisms that are existing in the population. You don't have mutants that do not produce the insulin receptor. You have mutants that have small changes in the sequence of the insulin receptor. You have mutants that change a little bit. These are called variants. You have haplotypes, and you have different haplotypes in the population, and you can study that in human populations. And so we can get the supercentenarians. In this case, we have Kalman here on the left with 121 years old. She died in the 90s, therefore there was not this massive genome sequencing efforts that we have now. So nobody actually sequenced the genome of Jean Kalman, but we can sequence the genome of other supercentenarians. These are all the studies that we were telling you about. And one very, very nice one correlated many of these variants. And so I wanted just to focus on this FOXO 380. This is one of the variants of FOXO that are present in the population. And what we can see here is that a certain type of variant, this increases the favorable gene. In this case, this variant increases in the people that reach the supercentenarian stage. Let's put it like that. People that reach 100 years old, they have a higher, in the population, there is a higher prevalence of this variant compared with the prevalence in the population which does not reach this 100. Look into the studies that we have with the model organisms, we can then correlate them with human studies and study things like this. Why? Because, of course, in humans, the only thing you can do is caloric restriction. Yeah, but that is, as you were saying, and with all the questions that you were asking, very complicated because we're touching many different things. Now, if we only touch a variant of FOXO3, we are only one of these transcription factors. The other thing is, what is this transcription factor then doing when it's in the nucleus? That's a lot more complicated. There's a lot of research which is being done nowadays, which has to do with all these different fronts that we have to look when we're looking at aging. Okay, so so here we have something which I think is very clear, and when we're doing the discussions of the papers, when we have no confinement developmental biology tutorials, is that I pick up papers from different subjects, so we can discuss them in the tutorial groups. And so of these, today we talked about only the DNA repair here and the caloric restriction, which in fact is not even here because it feeds into different things. But what we have here represented is a young man and an old man, and then all the things that can contribute to aging. There's many different cartoons of these with different things. In this case, it tells us about DNA repair, but of course, don't think that this is in this order, that, first you have a defect in DNA repair and then you end up having problems in mitochondria later on. No, it's not that. It's just a way of representing it. So things that can lead to aging: defects in DNA repair, telomeres, and telomeres, and we will very quickly go into telomeres now. Epigenetic modifications that I am not going to even talk about. Protein folding or proteostasis, there's lots of studies being done in protein folding, proteostasis in aging. Mitochondrial function, and I'll very briefly also talk about mitochondria. Stem cells, which is another world in aging that we could be talking about for a very long time. Cellular communication, which is a new way of looking at aging as well. So how do cells, through factors that are secreted, communicate with each other? So if you have a group of senescent cells, are these going to secrete factors that tell the other cells that these cells are senescent, for instance? And if they are young cells, will they secrete factors different from the ones that are senescent? And so that are now being done in organisms, not just in cell culture. Okay. And so very briefly, telomeres, because everybody that talks about aging talks about telomeres. And telomeres, the importance of telomeres, I don't have to explain that to you. We know that at the ends of chromosomes, we have telomeres. The shorter the telomeres, then the shorter the lifespan. Because as cells are dividing, telomeres get shorter and shorter. So what is this reminding us of? Of the Hayflick limit. So one of the justifications for the Hayflick limit is the fact that telomeres get shorter and shorter, and these cells will enter senescence because their telomeres are much shorter. Okay, so we have telomeres, and when it was found, it was the secret to long life. So you have long telomeres, you're going to live forever. You have short telomeres, you're doomed and you're going to live. And of course, that's not like that. And of course, the progress of this talk today has taken us into this. And if we think about telomeres, in order for telomeres to have an effect, you have to have a dividing cell. So if cells are dividing, telomeres have an influence in entering senescence, but if cells are not dividing. So most of the studies, or many of the studies that are done now with telomeres are done in stem cells, okay? So what happens with telomere length? Okay, so for dividing cells as we age, so we have telomere length here in human blood cells, and you have, from when we're babies until we're 65, and telomere length declines in dividing cells. I mean, nobody's saying that telomeres don't work. Of course, telomeres shorten as cells divide. But there are many problems. We have cells in our bodies, and the ones that are affected very highly with chemotherapy, for instance, the ones that are dividing a lot, like the gut creep cells, they will divide up to 5,000 times in humans, okay? And so they divide up to 5,000 times because they're results of stem cells, and they keep rebuilding the same population of cells. We also know that most short-lived animals have very long telomeres. For instance, mice have much longer telomeres than humans, and they live much less. And so the number of times that their cells divide does not correlate with the lifespan. And again, we have cells that don't divide that also lose function with old age. And this is one of the important problems with neurodegeneration. Neurons are not dividing, and they're losing function with age. So the whole problem with aging cannot be just telomeres. Of course, telomeres is a part of aging, but not the whole story. So it's not the holy grail as people thought at the beginning. There's also some human rare diseases where telomerase is effective. One of them is called dyskeratosis congenita, and it's a bone marrow disease because, of course, in the bone marrow, you have lots of stem cells and you have problems in division if you have mutations in the telomerase. But these people that suffer from dyskeratosis congenita and have bone marrow disease, this is not a progeroid syndrome. So these people are not aging faster. So you can have mutations in telomerase and not age faster. Mutations in telomerase that are implicated, of course, in not being able to maintain the telomeres, and therefore these people have shorter telomeres in their cells, but they don't have any progeroid syndrome. They don't have any aging syndromes. We have some of these progeroid syndrome cells, like the ones in DNA repair, that they have shorter telomeres, but this has been attributed to the failures in DNA repair and nothing to do with the telomerase. Okay, so then we move on from telomeres to mitochondria, which is the last part of this seminar. And mitochondria are important because oxidative stress has also been not just associated with aging, to do with DNA repair, but also with age. So not just with the DNA repair, but also with aging. And this came from the free radical theory of aging that said that organisms age because cells accumulate free radicals over time. So we have reactive oxygen species. These accumulate in the cells over time. This makes cells age faster. And this led to another theory, which is the mitochondrial theory of aging, that said that mitochondria produce reactive oxygen species and are the main propagators of free radical aging, damage, which meant that mitochondria could be one of the secrets and one of the agents that are involved in aging. And so this is also one of the fronts of research that we have now, how mitochondria affect aging. So we have mitochondrial health, and of course, how can mitochondria affect aging? By many different ways again, because of course mitochondria are also involved partly in nutrient sensing pathways. You have caloric restriction and you have physical activity, and that feeds into mitochondrial health. You also have mitochondrial metabolism, like mitochondrial fusion and fission, so the way that mitochondria fuse and then they divide again, and this is also a very important process for the energy metabolism of the cell. And then, of course, mitochondria are important for stem cell function, for senescence, and for many different things. And so all of these are studies that are now being done according to aging. We've been looking in many different studies now at mitochondria as important players in all the aging process. Also, we know, and this is also why it's so attractive to study mitochondria, that most aging-related diseases, in particular neurodegenerative diseases, have a mitochondria involvement. So in many neurodegenerative diseases, there have been found defects in the mitochondria of neurons, for instance. And then we also know that mutations in DNA repair genes can lead to mitochondrial dysfunction. For instance, in some mutants that are mutant for DNA repair proteins like XPA and CSB, the cocaine syndrome protein, these mutations have also been associated with mitochondrial dysfunction. And now here, you have to be aware there are two different things. One is that mitochondria have their own genome, and the other one is that mitochondria are not just regulated by their own genome, but they're also regulated by the nuclear genome of the cell. And so you can have proteins that are in the nucleus of the cell, which then affect mitochondrial health, and you can have the whole mitochondrial genome, which can also be affected and also has to be repaired, and etc., etc., etc. So how can we tackle aging? And this is I think the last slide that I have. Well, we can do it in many different ways, and we spoke about some of them here today, but of course we have a lot more. And as you see here, one of the main ones is dietary restriction, the insulin receptor, mTOR inhibition, and all of these are being studied in ways to be able to increase lifespan. We have mitochondria, and also one part which is quite important, which is removing from organisms senescent cells because of this intercellular communication. We have stem cell-based therapies, so what if we can just, using stem cells, renew the tissues in the body? We have rejuvenation factors also to do with intercellular communication. There are many studies where your blood from young individuals, for instance, from young mice, is being put into older mice to see if the cells and secreted factors from a young individual can then change the parameters of aging or lifespan of older individuals. So there are many studies being done that have to do with rejuvenation factors that are present in the blood. Then you have all the DNA repair and telomere part of aging, and of course epigenetics, and the way of looking at the chaperones and proteostasis and the way proteins are folded, which also is involved in the process of aging. So there's many different fronts. And then of course, each lab is looking at one simple thing that is related to aging, and then we have a whole picture which is very, very complicated. So your take-home message today is that aging is a very complex process regulated by environmental and genetic mechanisms, and we do aging studies to be able to know more about why we age, to know more about the genetics of aging, to increase lifespan, but mainly at the moment to be able to increase this health span, to make individuals healthier for longer. So I have my last slide here is with the task that you have. This task is put in the slides, but you also have it in the place where you have to put your abstract. So you have to do an abstract responding to one question that I'm putting you here. So basically, this is DNA repair failure, as we were seeing at the beginning of this seminar, is intrinsically connected with premature aging and cellular senescence. Patients belonging to xeroderma pigmentosum group A and cocaine syndromes group A and B, so one of the ones that we were talking about, cocaine syndrome, and another one with another syndrome, which is also DNA repair, and is also based on this nucleotide excision repair pathway that I showed you, they have defects in nucleotide excision repair. So these proteins are involved in nucleotide excision repair, but they also have early onset neurodegeneration associated with these phenotypes of premature aging that you saw in these children and teenagers. And so I would like you to discuss one possible hypothesis for the link between the involvement of these DNA repair proteins and in neurodegeneration and aging. And I have to tell you, there is not a right or wrong answer here. So any hypothesis that you can come up, which is well formulated and based on the data that exists, is good enough. And so the idea is that, in fact, the best thing is to have each group have a different hypothesis, because there are, at the moment, currently, many different hypotheses to justify or to correlate the phenotypes of DNA in the DNA repair proteins in the neurodegeneration and the progeroid syndromes that these people present. So with this, I finish. If you have any questions, I am still here. And if you then have any questions later on, you have by email. And I'm looking forward to receiving all your abstracts about this neurodegeneration and DNA repair link. Any questions? If you want, you can also ask me questions in Spanish and in Catalan. You don't have to. You can do, yeah, and the abstract, I mean, you can do it in English or in Spanish or in Catalan as well. I know this is to be published, but I asked any of the 3 languages. Okay, so do patients with altered NER pathway also have a great guess? Aha, a very interesting question. So some of them do, and some of them don't. So just to answer your question correctly, if you have people, for instance, with nucleotide excision repair defects, and they have this xeroderma pigmentosum syndrome, people with xeroderma pigmentosum, most of them will have a higher incidence of skin cancer, because the nucleotide excision repair pathway is one of the ones that deals with the removal of lesions that are made by a UV light. Okay, so if you have UV light and then the skin, you don't have DNA repair, and then you get skin cancer. These people have a higher incidence of skin cancer. Some of them will have neurodegeneration, and some of them will not. Okay, so in xeroderma pigmentosum group, they have all a higher incidence of skin cancer, and then some of them will have neurodegeneration and some will not. I will answer the next question when I finish this. Then we have cocaine syndrome. In cocaine syndrome, people have defects in nucleotide excision repair the same way, and most of them will have neurodegeneration, but they do not have higher incidence of cancer. Okay, so this is 2 main differences, and this is why it's so interesting to study these 2 different syndromes, because they affect the same group of proteins that are involved in one specific DNA repair pathway, but one of them is involved in the cancer-prone syndrome, and the other ones are involved in a disease which is not cancer-prone. Okay, I hope that answered your question. Now, I was asking, I know, that is a question that is, I cannot answer that with a yes or a no. Okay, so you have, how does epigenetics work? You have different regulation of different genes, okay? So you have different complexes and different ways of methylating and different ways of silencing parts of the genome, which are involved in the regulation of the genome. And so epigenetics is involved in regulating. Some of them are going to be more open, some of them are going to be more closed. And so it cannot be the similar mechanisms as mutations in repairing genes. It's actually a lot more complicated. You can have differences methylating parts of the genome that will affect aging. And so if there are some, in fact, with Drosophila, there are some studies where if you have differences in methylating some histones, you will have longer or shorter lifespans. But it's a lot more complicated than just you mutate this and you're going to get neurodegeneration or that. So reactive oxygen species and shorter telomeres can affect the aging process. Okay. How does the organism know which one of these problems to develop? Well, you have, it's back to that thing that we, Valentina, it's back to the to what we were discussing before. So if you have dividing cells and non-dividing cells, okay? So if we're dealing with aging, and this is why I made this big point about asking you, what do you think the majority of your cells are doing at the moment, is because if you're thinking about dividing cells, you're immediately, or at least generally, thinking about cancer. So let's say telomeres and reactive oxygen species involved in cancer is because you're dealing with dividing cells. if you're talking about reactive oxygen species and telomeres in non-dividing cells, in fact, telomeres cannot really do very much if a cell is not dividing. So in fact, that can be regulating something else, but not the division of the cells if they're not dividing. And reactive oxygen species, they can do many things. One of them is to actually damage the DNA. But if you are a cell which is not dividing, you're not creating mutations, okay? So the organism does not know which problem to develop. The differences lie on the fact that in order to produce a tumor, you need cell division, okay? And in order to age, in fact, you have some part of the body which is connected with cell divisions, but some others that are not. Okay. Any more questions? It is enough with the information that you have given us. You need to, I mean, if you want to do, I mean, you can do it. You can do it. This is why I also put this in at the end and explained you a little bit more. You can do it with the information. So you can come up with a hypothesis with the amount of theoretical knowledge that I gave you. But of course, if you want to do a good job, you should search. Why? Because if you quickly do a search, you will see that there are many different hypotheses related with the link between DNA repair and neurodegeneration. But of course, you're free to think about it without having to search. You don't have to go and do a big bibliographic search to write a big research project. No, all I want is you to think about hypotheses that will explain why these people have DNA repair mutations in DNA repair proteins, and at the same time, they develop neurodegeneration. Because remember, when we're talking about neurodegeneration, we're talking about cells that are not dividing. And so the DNA, we have to be talking about more the DNA damage, obviously, than any mutation. You're welcome. So you think you have until Monday to do this? And I will be looking forward to reading your, if you have good hypotheses, then I will ask you to come to the lab, because we have, in fact, some projects that have to do with this, with DNA repair and the development of neurodegeneration later in life. Okay, if you don't have any more questions, we can say goodbye.